education purposes. They also provide revenue to the college and are a big source of recreation for our local communities. Located on the east side of the Coast Range, they're drier than most of the rest of the Coast Range, given they're in the rain shadow. So they have mild, wet winters, but also hot and dry summers. Historically, the Kalapuya people use extensive burning to maintain oak woodlands in this area, with small reserves of Douglas-fir and some of the draws. But with their forcible removal, the landscape has shifted to a Douglas-fir dominated forest with fire suppression in the area. The mature forest study was implemented both on the McDonald and the Blodgett tracts, but my research looks at only the McDonald stands. And it was implemented in 1993, kind of at this time in Pacific Northwest forestry, when habitat, particularly for rare species, was becoming increasingly important and talked about as a social value, and timber as it had been came under fire. So started looking at how to extend forest rotations and create a more heterogeneous structure. To this end, there were 4 intensities of thinning implemented in 2 different spatial arrangements, and these were replicated in 3 blocks on each of the forests. In 1994, these were underplanted with Douglas-fir, western red cedar, grand fir, and western hemlock, and 3 levels of herbicide application were also applied to the understory vegetation. However, this replanting and herbicide does not really affect the overstory systems that I'll talk about. And so we have these 4 really intensities of thinning that are important to think about, in addition to the structural differences in the gap thin. To complicate it, the medium and medium highs were rethinned 8 years after the original thinning, which kind of put them more in the densities of the light thin in the end. So getting more into this uniform versus gap, variable density thinning at this time was a big process coming to be thought about as a silvicultural system that can be really fine-tuned to your landscape to preserve certain elements, or but also keep timber production going. It uses gaps, skips, and matrix thinnings to create this different structure, as you can see in the image. A very heterogeneous structure that hopefully produces both wildlife habitat and can still produce timber. In contrast to other thinning methods, VDT can promote regeneration. So overlaying this with this idea of drought and increasing drought that we will see in the Northwest, we use PDSI as our metrics for choosing drought years to study, and using a -2, which corresponds to a moderate drought score. And using mean annual PDSI values, we chose 2001, 15, and 16 as years of interest. Because the drought didn't abate between 2015 and 16, we lumped those 2 years together into a single drought event. So a couple definitions regarding our response variables for this study. Resistance is a tree or stand's ability to continue growing at its pre-drought rate during a drought event. So really trying to figure out if during the drought a tree can't grow at its normal rate. And resilience is a tree or stand's ability to return to a pre-drought growth rate after a drought event. So really, does the drought continue to affect the tree after the drought itself has ended? So for our field methods, in summer 2020, myself and a field technician collected data on the MacDone. We put in 5 sample points per subplot. They were randomly assigned, and we used a nested circle design in which the inner circle was used to determine which trees to core. We only cored Douglas-fir, which was the dominant overstory, and anything above 10 centimeters. We also took a variety of measurements, including heights, DBH, live crown height, crown class, and any damage. Primarily ice damage was the dominant one in our stands. And a larger circle was implemented in which DBH and crown class and species were recorded to establish and fine-tune a competition level of these individual core trees. In the fall, I switched over to the dendrochronology side of it. Each core is mounted and sanded. I initially hand dated each one and then measured each annual growth ring, which were then cross-dated using Cofetcha. And this is all pretty standard dendrochronology methods. For our analysis, I used a combination of allometric equations and the ring width data to calculate basal area increment, which is pretty much how much area the tree grew if you looked at a cross-section of it at breast height. Yeah, so it's very closely related to basal area, but it's looking at that yearly growth. And we calculated resistance and resilience scores for each individual tree from those BAIs, and then used mixed linear models to analyze differences according to treatments and their influence on our response variables. So looking at our individual tree response, these models included all of the tree measurement covariates, and all of my figures have pretty much the same XY axis, with treatments across the x-axis going from our lowest density to our highest density. And then our y-axis has the mean response variable. In this case, it's the estimated marginal means. And just a couple sort of measurements on there to highlight. The one, a score of 1 means that nothing really affected the tree's growth, or the drought didn't affect it. It grew as much as it did before the drought event. A 0.75 is a pretty significant effect from disturbance ecology, that drop in 25% of growth or 25% mortality or anything like that is kind of a big estimate that something large has happened. So here we can see in 2001 at the tree level, not much has really affected them. The high densities are doing a little bit worse, but there's really no significant difference by treatment. And in 2015 and 16, at first glance, these look pretty similar, but you'll notice that the means are a lot lower. Some of them are dipping below that 0.75. But once again, there's no real difference by treatment. It's just overall lower than the previous at the individual tree response. And this is both for resistance and resilience. Once we get into the stand level, though, it starts becoming, the differences are bigger. And here we are able to compare both our first and second response droughts. So our first response, or drought, sorry, is in red and the second in blue. And we can see that there's quite a difference between the two droughts, both for resistance and resilience. We believe this has a lot to do with the fact that the second drought was both longer. It was a 2-year drought. It was hotter, which puts an extra physiological stress on our trees. And also it has been longer since that initial thinning. So the competition has increased by the second drought. So this is kind of just the preliminary data of all this research, where some of the questions we're going to start looking at now is really trying to tease out why there's a difference in the stand response and the individual tree response. And additionally, we're working up some volume calculations to work on trade-offs for the timber side of this and really understand, is there a difference in standing volume and therefore economic differences in the different treatments? Some of the data suggests there's slight differences in gap and uniform responses, so we're going to investigate that further. And I haven't really mentioned it, but we did collect data on the understory red cedar and western hemlock and try to compare if there's a difference in their drought response within the gaps or in the matrix. And so a couple of key takeaways are that intensity of drought seems to have a huge effect on drought response and time since thinning. And both of these seem to override any influence of the thinning intensity. And that's about it. Thanks for coming, and thanks to everybody who's helped me with this project.
Great, thanks, Madeline. If anyone has questions for Madeline, we've got plenty of time, so either put those in the chat or raise your hand. See, you've got a question from Todd West here, just asking where the plots in the MacDun that you used are.
So the mature forest study is located kind of between PVR Arboretum and Lewisburg Saddle, kind of right in the middle up on that ridge. You'll know them when you see a lot of blackberry.
I was wondering, this is a pretty local study, and I was kind of curious if the results that you're finding seem to line up with similar studies, or if there is similar literature out there about this, like drought responses across silvicultural treatments and what you expected to find.
Most of the studies are from wetter sites on the Coast Range or even on the east side. And I think they support more that there is a difference in thinning intensity with this stuff, but the time since treatment, our stuff is aligning with. So yeah, I think it'll be interesting. This is a very dry, kind of unique site for this research because there's not a ton of dry Coast Range sites out there.
See, we've got a question from Dave Shaw here. What do you think about thinning and drought in general?
In general, I think it's helpful from my reading in the research. I think there's a line when, especially for understory plants, it can be too much. If you start getting too much sunlight down through the canopy, it kind of raises temperatures and can shift back to not being helpful, but a certain amount seems across the research to be helpful.
We have a question on here from Meg Krawczyk. It looked like the high gap response was lower than others in the first drought, but higher than others for the second. Is that kind of what you saw, and what do you think was going on there?
Yeah, it is. Some of these numbers are not super consistent across the board. Looking at it, there's a lot more suppressed trees in the high gap, which are potentially playing with our numbers a little bit because their growth rates just kind of plateaued at a certain point. And so we're gonna spend a little more time teasing that apart, but particularly the high gap, the matrix are pretty dense. So there's a lot more suppressed trees in that area.
And from Amanda Brackett, do you think there's a threshold value in drought or water availability below which thinning is no longer beneficial?
Yeah, I mean, I don't quite know where it is, but probably. I think we've seen a good bit of mortality, not in my stands, but on other stands of the MacDun. And I know some of the other presenters and posters are actually looking at this question and trying to figure out a lot of more water and physiological aspects that go into that, specifically on the MacDun.
Great. I think that's probably it for questions. So thanks, Madeline. That was great. And we have about 5 minutes until our next talk. So I don't want to jump ahead. If you have a few minutes to kill, feel free to go on and leave feedback for Madeline on the Qualtrics forum online. Otherwise, we'll just sit tight.
Thanks so much.
All right, Stephanie, if you want to share your screen, I think we can get going. All right, that's looking good. All right, so,
Sorry.
it's okay.
Oh, do you see the presenter view or like the notes section?
I just see the slide, it looks good.
Okay, cool, thanks.
All right, so next up we have Stephanie Winters, also in the Department of Forest Engineering and Resource Management, here to talk to us about exploring soil carbon dynamics using a common forest organic matter removal experiment from Idaho to New Zealand. And yeah, take it away, Stephanie.
Thanks. Yeah, I'm Stephanie, and I would, I would just like to say that, yeah, people's presentations today are some of the best presentations I've ever seen. So you guys, like, very good work. Okay, yep, so this is my title, Persistence or Resilience in Soil Carbon Pools. The question, explore, yep, okay, so globally, more than 40% of terrestrial organic carbon is stored in forest soils. So on the earth, 40% of our terrestrial carbon is stored in forest soils. And in temperate forests, so what we have, 60% of that total ecosystem carbon is stored, once again, in forest soils. So this Meyer et al. TAM review, it's just a meta-analysis on forest management activities on soil organic carbon stocks. So this came from that paper. And so they have a visual that would help us kind of explore what that looks like. So this is what it looks like. So on the top, we have vegetation, and that's not just above ground vegetation. That's roots included in there. And we can see that most of that carbon, again, 60% is stored in either subsoil or topsoil of the forest, in the forest soils. And so my roommate Lucie asked, okay, but like, why does this matter? And that was a really good question, because it's like, why does it matter? And it matters because we have so much carbon stored in these soils that even a small percent, if it's lost, could have huge negative consequences. It might not actually matter to that percent, like the overall percent might not decrease, but it could have huge negative consequences on increased respiration by microbes. If that's the way the carbon is lost, increase in CO2 into our atmosphere, creating a positive feedback, as well as methane. And also, because we're foresters, we think about productivity. And over the long term, if we lose carbon in our soils, there's a huge potential that we'll also lose productivity. If we're losing soil organic matter, we're also losing those other nutrients associated with soil organic matter. So what is soil organic carbon? Well, this is soil organic carbon. It's a lot of dead stuff. It's just living things being transformed into dead things. You have leaf litter, you have char, you have roots, and you have biological critters. And so we have cellulose and lignin. These are carbon molecules, carbohydrates that are coming from this leaf litter. You have pyrogenic carbon that is produced when wood is charred. And this is from Catherine McCool's site, a master's student in our lab. And you also have lipids, and you have root exudates. You have microbiota and macrobiota exudates as well. So when we talk about soil organic carbon, visually we see it all the time, but what we don't see is the decomposed molecular state. And so previously people thought that soil organic carbon persisted in the soil for so long because once it started decomposing, it actually started forming these really big, huge, complex molecules, what people call recalcitrant carbon. And they were so complex that microbes could not decompose them with their enzymes. But what we really found, well, I didn't find this out, but what was discovered is that these complex molecules are actually created from the lab methodologies themselves. And what we're really finding in soil are these carbohydrates, these lipids that exist. But why are they so persistent in soil? And so that's this new paradigm shift where it's not, they're so complex that they cannot be degraded by microbes, but that they're forming a bond with, they're protected by minerals, association with minerals, organo-mineral complexes. And so I like this scheme, and I did have a citation, it did not copy over. But so in soil, we have minerals and we have these carbon compounds, and minerals are positively charged. And when carbon compounds start decomposing, they're oxidized and they have these negative hydroxyl groups. And so you can see, I don't know if you can see well, anyways, you can see that the organic compounds are absorbing because of charge onto these minerals. And then those minerals start, coming together. And what you have is a soil aggregate. So when you're in the soil and you can see little aggregates, it's essentially these organo-mineral complexes. And then we can see that they just form bigger aggregates, and the brown, or the carbon, is protected in these little aggregates. So microbes can't actually access that carbon. So that's this new paradigm that we're working with, that we are exploring for my dissertation. So the overall objective is to elucidate the mechanisms that impart persistence and resilience to soil organic carbon after extreme disturbances in forest soils around the globe. And my research question recently has been, can we use the parent material soil mineralogy to predict how soil organic carbon will behave after an extreme disturbance over time? And so my hypothesis is that we will see trends in these pools, the total carbon amount, the age, and we can age it with radiocarbon data, and also the composition. So the different carbon molecules from different parts of the plants, whether it's above ground or below ground, over time. And this is going to be dictated by soil mineralogy. So going back to that organo-mineral complexes. So proposed impacts, like, why are we even doing this? Cool. We find the mechanisms, but what does it matter? So hopefully this will impact long-term management of temperate forest soils. If you're a soil scientist, or you're an ecologist, or a timber manager, and you work for the government or whatever, you can have an idea of how your soils are going to behave after you do a timber harvest or after there's a wildfire. So you can plan in the future to maybe not do that harvest, or yeah, we can harvest and we're not going to lose carbon. And so also it's going to be across different soils and forest types along a climatic gradient. So we'll get a good profile of that, and also just tying back, identify vulnerable and resilient forest soils to carbon loss. So my sites, I'm using these Forest Service long-term soil productivity sites that were established in the late 80s by Bob Powers. And now these sites, the Forest Service sites, but now it's global, and New Zealand also has sites. There were 2 organic matter removal treatments that we're studying in these experimental designs. Here are the sites I'm focusing on today in California, Oregon, and Idaho, and also the northern island of New Zealand. And here's just a brief picture of the organic matter removal treatments that we're comparing at each site. So just 1, or organic matter removal 0, the stem is just removed, pretty traditional, and organic matter 2 removal, where everything is removed, and also the forest floor. So we're bookend between, pretty minor to pretty extreme forms of organic matter removal. And I just wanted to start the results section with this quote. Many ecologists glibly designate soil as the abiotic environment of plants, a phrase that gives me the creeps. And this is like the soil science daddy, like started soil science. Everybody knows him. So a living view of soil recognizes the fundamental role of organic molecules in shaping soil chemistry, that soils are very dynamic and subject to both harm and improvement. And so I wanted to show everybody these soil profiles from my sites in the United States. And what you're looking at is the original soil series, the official soil series description of that soil at my sites. And we can see they differ in depth, they differ in color, which could be an indication of mineralogy or organic matter, climate, relief even. And so they're really diverse. Each site, almost every site has a different soil profile or soil series. And then I thought, okay, let's break it up by soil order. We have alpha soils, which are high in clay. We have andisols that originate from volcanoes. And we also have inceptisols that are relatively recent soils, as well as ultisols, Oregon State soil is an ultisol, Jory, which is highly weathered and also has a high accumulation of clay. So we can tell that these soils have different environmental factors working and different parent materials as well. Okay, so today I'm focusing on these 3 soils. The sites are Blodgett and Lowell Hill, Central, and Brandy City. And then 2 New Zealand sites, but I don't have those profiles yet. So this is a graph of all of my sites, and the 0 line indicates effect size. If the point is above the line, then these sites have actually gained soil organic carbon from pre-harvest. And if it's below, these sites have lost soil organic carbon after harvest. And on the x-axis is years post-harvest. And so we can see that the soils are not really breaking up by harvest treatment. Like they stay relatively similar as the years progress, which was surprising to me. But we can see that below we have our sites. The Kinlaith in New Zealand and Tarawera in New Zealand has lost quite a bit of carbon after harvest. So these are like our poster children for our definitions of persistent, resilient, and vulnerable soils to soil organic carbon. In Central, we can see a steady, relatively steady, it's not losing carbon, it's actually increasing slightly, whereas Blodgett is resilient. It loses carbon 10 years after harvest, but then it regains it 20 years, compared to our vulnerable soil, Tarawera, which just steadily decreases with years. So going back to productivity, we can see the same trend in total nitrogen and when we are thinking about productivity, we think about nitrogen often because trees need nitrogen to produce leaves, for photosynthesis, and it's following that same trend. So if we're losing carbon, chances are 25 years down the line, we also might be losing nitrogen as well. And so this is, shout out to Ariel because, we're all using the code and the stats we learned in her class. This is a linear mixed effect model result. And on the x-axis is our total organic carbon, and the points represent our estimated means. The line represents, again, if it's below 0, it's lost carbon. And the gray represents, a significant or practical amount, well, I guess, a negative effect if it were to lose 1%, if it's in the gray. And we can see that the soils whose parent material is granodiorite and volcanic mud flow, those sites over time significantly have increased in total organic carbon, whereas the basaltic tephra in New Zealand, so that's volcanic. Oh my gosh, I'm almost out of time. Okay. Anyways, wow. Okay, yes, so we can see that parent material in time is significant to total organic carbon loss. Okay. Anyways, would like to thank our Forest Soils Lab and the Forest Service, and my think tank that I'm a founding member of with Lucie. So thank you, everybody.
Thanks, Stephanie.
Thanks.
You can,
sorry it was so chaotic.
It's okay. If you want to spend another minute or two there, you're welcome to. Got a little bit of time. But
you know, I could just chat. People
Okay.
have questions.
Yeah.
Cool.
Let's see. Yeah, Rebecca.
Okay. I was typing out a question, but I figured I can go ahead and say it out loud. So I have two questions, I guess, and I'm not a soils person, so I'm sorry if some of the other though covers it, but I was curious whether you're using any ultramafic soils like serpentine derived soils.
Yeah, I'm not, but man, if I was, that would definitely be, you know, every soil is really unique based on the chemistry, depth, mineralogy, just like it's indicative of the climate it is in, you know, and that's going to tell you a lot about the vegetation also potentially. So yeah, like with your serpentine soils, your vegetation is going to be very specific. Man, I don't want to deal with that. That would be such an outlier for many things, I'm sure. Very cool soils though.
Mark Kirsten's asked in the chat, why New Zealand?
Good question, Mark. Well, New Zealand started those they have long-term soil productivity sites, and Jeff is working with Scion. It's like their Crown Royal Institute that they have there. And they have 30-year, 25-year-old LTSP sites. So they have this archival soils also, and it's like a temperate forest, and they grow a lot of Douglas-firs and Pinus radiata too. So we can compare those sites with the United States.
See, we have a question from Doug Turk in the chat. For sites that are losing carbon and nitrogen, do you have any ideas for why that's the case?
As far as?
Yeah.
So with those soils, I believe, I didn't really go into it, but the soils that are losing it, they're volcanic. The mineralogy is tephra. It's really glassy. It's low in clay. So they don't have a lot of charges potentially to hold on to those, to that organic matter. Whereas the soils that are higher in clay, which I skipped through, apologies, we see that there's more organic matter, and that's in the literature too.
And it looks like Becca had a follow-up here, asking whether or not you looked at the microbe communities and the different soils that you were looking at.
No way. I did that on my master's. That's super hard. And we're using like 30-year-old soils. So, you have to have that soil freeze dried or, put in the freezer. And this is just like air dried for many years in Forest Service warehouses. But good question. That would be cool. Yes, of course. That would be awesome.
And Mindy asks, was there a relationship between the carbon loss and harvest activity, or did it vary only by soil category?
For that model, I did not look at harvest treatment to keep it, relatively simple, because prior to that, not as complex models, I guess, or didn't take into account variability. I wasn't seeing big differences between these two different harvest treatments. And my lab mate Adrian, we talk about this, and it's like maybe harvesting is just such a huge shock to the system, to soil, that even just removing the forest floor isn't going to change how carbon behaves. And so that's another hypothesis. Maybe it's more root dominated carbon than litter dominated.
And then a final question, somebody asks, when and where can they find your published paper on this?
Is that C? Or wait, who? Anyway, I don't know. Maybe stay tuned. Thanks, everybody.
Awesome. Thanks, Stephanie. Let's see. Lucy, do you want to share your screen?
Yes. I just stopped the recording. Okay. Is it looking normal?
Yeah, it looks great.
Cool. Let me just move my Zoom toolbar.
Yeah.
Okay.
All right. So next up, we have Lucie Law, also from the Department of Forest Engineering, Resources, and Management. And Lucie was also one of the key members who organized WFCRS this year. And she's here to tell us about riparian restoration. So take it away, Lucie.
Thanks, Graham. All right. So hello, everyone. Thank you for attending. My name is Lucie Law. I'm a master's student working with Dr. John Souter. And today I'll be talking about our research studying the planted tree survival and growth within riparian restoration areas. Okay, so here's the presentation roadmap. I'll provide some background on riparian areas and restoration, talk about the study design and data collection, go over some survival and growth analysis, impacts of this research, and then hopefully we'll have some time to chat. So what are riparian areas? These are areas that are the interface between aquatic and terrestrial habitats. In this area, there is a lot of interaction between the stream channel and the vegetation on the stream bank. Billions of dollars are spent on riparian restoration because of the important ecological functions this ecosystem provides. Specifically, the canopy from the trees and the shrubs can limit the amount of solar radiation reaching the stream, thereby cooling the water temperature. The roots from the vegetation bind the soil together and reduce bank erosion, and this can reduce the amount of sedimentation in the water, improving aquatic habitat and water quality. Additionally, as the trees grow and eventually die, they can be a source of large wood, which provides habitat and channel complexity. These functions improve water quality, and in Oregon, they're especially beneficial to the salmon and other fish in our streams and rivers. So for my study, I'm using data from 13 projects that were planted by the Coos Watershed Association beginning in 2002 through 2009, and they were planted throughout the Coos Bay watershed. The project goals were to improve the ecological function of the riparian areas, but also to monitor the plants long-term. So monitoring began in 2002 with the first project, and we finished the most recent round of sampling in summer 2020. Will there be beyond? Only time will tell. So this is a very exciting research opportunity because typically riparian restoration projects are only monitored the first couple years after implementation due to funding and staffing limitations. So here's a picture, an aerial imagery of what where the projects are located on the landscape. Most of the projects were planted on industrial logging properties or agricultural landscapes. And in each project, I have 3 here, they're defined by different color dots. But in each project, the total project area, 10% of it was designated as long-term monitoring plots. And each planted tree and shrub within that monitoring plot was individually tagged so that it could be tracked through time and measured for height, DBH starting in 2012, levels of competition, damages, etc. Okay, so before I go into the details of the research, I'd just like to take you on a riparian restoration journey. This is a private lands project. The property used is for livestock grazing, and it's along the Millicoma River. So the initial conditions in 2008 are shown here. You can see that the riparian area is dominated by Himalayan blackberry There are very few trees providing shade and cover over the stream. So the first step after you're on board with the landowner and you have a plan is to prep the site. This included just mechanical brushing and removal of the Himalayan blackberry. A few months later, the project is planted and the trees and shrubs have added protection of weed mats and Vexar tree tubes, as well as bamboo stakes. All of these are just intended to reduce the competition of grasses or brush, reduce browsing. Additionally, trees with protection like this are a lot easier to find when you're coming back to repeat monitor them once the vegetation grows up around them. And there was also a fence installed to exclude the livestock from this riparian area. So 2 years later, this is a picture from a maintenance mission, and maintenance included just weed whacking or mowing the brush and grass around the trees. And this is from one of the monitoring plots, and you can see on this plot T-post that there are tags already collected from trees that have died. Okay, and here is monitoring in 2013. The trees are about 5 years old at this point. We still have a lot more tags on the plot T-post, but I'd like to draw your attention to just like how open it is still. You can see across the river to the road, but take note of the Sitka spruce and these cedars, because the next slide is from monitoring in 2020. Okay, ready? Wow. So this is monitoring from 2020. It's the same plot. Here's our Sitka spruce. Here's a cedar. It's totally filled in. You can't see the road. And I just really want to emphasize here that these projects take a lot of time and a lot of effort. Depending on the project's long-term goals, it can take decades for the benefits of these plantings to be realized on the ground. So it's really important to understand what these trees and plants are doing out there. Are they living? What's happening? So to just recap and give you a overview of the data I have, we have 13 projects that were planted from 2002 to 2009. We have 160 permanent plots and almost 5,000 uniquely tagged trees. And over the years, we've compiled 35,000 sampling records. And just generally out of that data, 32% of the trees and shrubs are presumed to be alive and 68% are presumed to be dead. And there's a little uncertainty there because it's always sometimes hard to find a tree or not, but I think it's pretty good. Okay, and so this chart is showing you the number of trees that were sampled 0 to 18 years after planting. And just due to like when the project was planting and just general funding and work limitations, logistics, the only year that all the trees were measured was at year 2. But you can tell that the vast majority of all the trees were sampled from 1 to 5 years after planting. The trees up here at 15 to 18 years after planting are from the 2 earliest projects that were planted in 2002 and 2003. So, just how time works, we can't have that many old trees yet. So with this data set, the research question I'm focusing on today is what factors affect planted tree survival and growth in these project areas. So I'll be looking at damages such as insect damage, beaver damage, and browsing, and stock type, plugs to the bare root. And so starting out really general still, this chart is showing the survival of all the species from all the projects from the most recent sampling event. So this chart has all of our trees and their age 12 to 18, again, depending on when they were planted. And this small number at the top of the bar indicates how many individuals of each species was planted. So for big leaf maple, 436 were planted, and we have about 25% survival. So you can see there were a lot of species planted, lots of variability in the number of individuals. But for the rest of the presentation, I'll just be focusing on the 3 most numerously planted conifer species, which are Sitka spruce, Western hemlock, and Western red cedar. So taking a closer look at the survival for these 3 species, I used a binomial generalized linear mixed model to estimate mean survival of the 3 species from initial planting, year 0, through the 5th year after planting. You can see that Sitka spruce and Western cedar are doing pretty good with estimated mean percent survival of 60% and 70% respectively. On the other hand, Western hemlock really took a nosedive year 1, and through year 5, the estimated mean survival is about 10%, with 95 confidence intervals ranging from 5 to 18% survival, mean percent survival. So when field crews are out sampling the trees, they would assess each tree and record and note if they had any damage. And they would record the damage type and the level. There are many damages such as browsing, insect trunk girdling, broken leaders, etc., but I've condensed them down into 5 categories of none, other, foliage, stem, and leader damage. So again, on the x-axis, you have years after planting, 0 being the initial planting, and then through growing through the 5th season. And then on the y-axis, you have percent of live trees with damage. And you can see that initially we're below 10% damage, but as time goes on, each species at least has about 40% of the live trees damaged at some point. So that leads me to the question of what is the difference on average of damaged tree height compared to non-damaged tree height. So again, I have my damage codes up here so you can understand better. 0 means no damage, 1 is light damage, 3 is moderate, and 2 is moderate, and 3 is heavy, meaning over 75% of the tree was affected by damage. And for this analysis, I use a linear mixed model with tree height as the response variable. And in each comparison, the mean height of trees with no damage was subtracted from the mean height of trees with damage. So a negative value, here's 0 right here, the dotted line. So a negative value indicates a decrease in mean height. For trees with damage level 3, check out Sitka spruce and Western red cedar. So trees with damage level 3 compared to non-damaged trees were estimated to have a shorter mean height. For Sitka spruce, mean height was estimated to decrease by about 0.65 meters, and Western red cedar mean height was estimated to decrease by 1.6 meters. So I need to talk about Western hemlock for a second. Western hemlock was the 3rd most numerously planted. However, by year 5, we only have about 10% survival. And that means we only have about 36 Western hemlock surviving till year 5. So the confidence intervals for this analysis are pretty wide because of the small sample size. And they're basically indicating that damaged Western hemlock could grow taller than a non-damaged Western hemlock, or it could grow much shorter. So there we go. And then the next step for this analysis would be to see if different damage types, such as the stem leader, foliage, affect the mean height in different ways. Okay, so how do odds of survival change with stock type? For this one, I use a binomial generalized linear model with a logit link. And the estimated proportion of trees of each stock type that survived was used as the response variable. The gray rectangle represents the practically relevant value of a 2.6-fold increase, and this increase in odds of survival could make stock choices driven by monetary value, as riparian projects often have tight budgets and practitioners may not want to plant a stock type that doesn't survive. So the odds ratio of survival for Sitka spruce bare root was estimated to be 6.6 times greater than plug stock at year 1, with the 95% confidence intervals ranging from 2.23 times higher to 19 times higher. So for spruce, there is evidence that bare root stock could have increased odds of survival compared to plug stock, but for western red cedar and hemlock, it doesn't seem to affect it. Okay, so the impacts of this research. The results could be directly applicable to agencies implementing riparian restoration projects throughout the Coast Range. These results can fill the knowledge gap of how planted species grow and survive in riparian areas. We could provide recommendations for species to achieve management goals. And overall, I just hope that this research can improve riparian restoration by continue the conversation of project design and species selection to achieve the long-term goals of a functioning ecosystem. So with that, I'll take any questions you may have, and if we're out of time, please email me.
Great, thanks, Lucie. We've got about 5 minutes for questions, so you can definitely do some questions. First one is in the chat from John Souter, asking if there's a relationship between stock type and damage rate.
There a difference between, sorry, can you say that again?
He says, is there a relationship between stock type and damages?
That's a good question. I haven't explored that yet. There's literally just so much data and so many things to look at. It's just every day there's something else, but I'm gonna write that down.
Similar question from Doug Turk. Oh, sorry, I'll let you write that down.
No, go for it. I'm good.
Similar question. What are the most significant types of damage to trees? Is it browsing, insects?
Yeah, well, that's one of the questions I'm gonna look at next, because, well, it's significant in which way? There are damages that happen quite often, but if you have a beaver damage, I would say that's pretty significant because it would chop down the whole tree. So yeah, I'm going to look at damage, types of damage, how often they occur, and the effect of survival.
A question from Mark Kirstens. Does shrub regeneration cause stress to the trees?
Cool. Yeah, well, so at the plots, we've only monitored the intentionally planted trees and shrubs that were tagged at initial project establishment. And we have recorded the competition of blackberry and grasses and other brushes. And that's definitely a question I will be exploring. The Himalayan blackberry in that region is wild, and it for sure has a detrimental effect.
And Steve Strauss asks, why did the hemlock do so badly? Any obvious reason?
Well, what I've been thinking about is hemlock is not generally what you might think of a pioneer species, one that is first to come in after a disturbance. So I think it is probably just not loving the exposed, freshly cleared riparian area. It would probably do better if we could plant it after a few years after some of the trees are growing taller.
Rebecca Weber says, great job, Lucie. Why do you think there is a difference between stock type and bare root for some species, but not others?
Yeah, that's interesting. Definitely need to explore that more. It was yeah, it's interesting that it matters for Sitka spruce, but maybe not for hemlock and cedar. Again, perhaps maybe hemlock, just because my sample size is kind of wacky. Sitka spruce, we have over 800 plants and they all had really good survival. So maybe it's just kind of like a sample size thing.
I guess Fitz points out in the chat here that hemlock is sensitive to planting stress generally.
Cool.
And then I think we have time for one more. Paul asks, would you clarify what your fixed and random variables were in your mixed models? And are you dealing with binomial counts or binary variables in your odds ratio tests?
Cool, Paul. Thanks for that question. I asked Stephanie if I should put those in my presentation. She's like, no. So that's why. Okay, my fixed effects were an interaction between species and stock type and time and ground. And my random effects were my project and plot. And I'm dealing with survival. So binary variables of 1 being survival and 0 being dead.
Great, thanks, Lucie. I think that's all the time we have. So I'm going to switch here to Sudeera. Would you mind sharing your screen when you have the chance?
Okay, can you see the screen, the slide that she had?
Yes, looks good.
Okay, cool.
Yeah, thanks. So next up, we have Sudeera Wickramarathna from also the Department of Forest Engineering, Resources, and Management, here to tell us about an assessment of herbicide effectiveness on reed canary grass. So take it away, Sudeera.
Okay, thank you. So hello, everyone. I'm Sudeera. Today, I'm going to present one of my research projects, assessment of herbicide effectiveness on reed canary grass. And this is a unmanned aerial system data processing case. This work was carried out with my advisor, Dr. Bodwin Strimbu and Dr. John Schauder. This presentation is focused on the remote sensing component of my project. So a little bit about the background. Earlier, reed canary grass had been cultivated for forage and used as a grass fodder for livestock. It grows as a dense monoculture, spread rapidly, and later become a major threat to many wetland ecosystems. So it requiring costly control measures, for example, mowing, grassing, or prescribed fires, or herbicide applications. Based on that, I would like to describe about my objectives of this research work, primarily identification of the most environmentally friendly method of restoring riparian areas impacted by reed canary grass using herbicides. Secondly, to develop a procedure that assess the efficiency of reed canary grass removal based on unmanned aerial systems. Restoration treatments, we have implemented 7 herbicide treatment based on the glyphosate, and we have used rodeo, that's the glyphosate agent, the competitor, highlight, and the water to make the herbicide treatments. In this experiment, we have used 5 replications. Also, this was a completely randomized design organized as a factorial experiment. The study area is located close to the Jackson Bottom Wetlands Preserve in Hillsboro, Oregon, and it covers around 38,000 square meters approximately. As you can see in the bottom left figure, we have a total of 38 plots distributed throughout the study area. Details about the data acquisition and processing, we have used DJI Matrix 210 as the UAS platform. Also, we have used 2 different sensors, including the multispectral sensor, that's slant range 3px, and the RBG sensor, that Senmuse X4S. For image processing and data analysis, we have used 3 different software packages, including Pixel 4D was used to create the orthomosaic ADAS image in 2020 for image enhancement and the analysis. And we use R for accuracy assessment purpose. Okay, discuss about the data analysis. In the study, we have used 3 different image pre-processing approaches, including the radiometric enhancement. This include noise and haze reduction, spatial enhancement, including 9-directional edge filter, creeps, and adaptive filter. Also, we perform Fourier transformation analysis as well. Third, spatial enhancement. Basically, we use this approach to create new bands out of the initial band. Also, I perform both K-mean and isodata unsupervised classification approaches, and the better results were obtained by K-mean. Also, I'm planning to do supervised classification like maximum likelihood or random forest in future. Now I would like to discuss about some of the important results in the study, as I mentioned in the previous slide. With the noise reduction approach, we could be able to preserve the fine details in the image, such as thin lines, while removing the noise along the edges. For example, if you see the image on left-hand side, that's before the processing, and the right-hand side is the after the processing. As you can see here, the result is the reed canary grass edges are more visible now compared to the left-hand side image. Shadows areas are more clear. If you look at some areas, probably initial image thought will say that's a shadow, but once we improve the image by using the noise reduction, that's not actually the shadow, but it's a part of the reed canary grass. Also, it helps to see the unhealthy and healthy reed canary grass. For example, here we assume that's a healthy, and if you see this area, that's the unhealthy reed canary grass. The color of the image is basically, I use false color composite to see the features very clearly. That's why I use different colors instead of the true colors. So also it enhances to look at the other vegetation types, and they are distinguishable. For example, if you look at this corner, there are different vegetation types along this line as well. But in left-hand side, we cannot see that much. Also, further, we have implemented haze and strip reduction technique. This allows us to correct the haziness or the reduced contrast in the original image. As you can see in the left-hand side, that's the original image. In the right-hand side, it's clear now, and you can see that most of the strips are removed. And if you look at this object in the left-hand side, that's the original one. It's not clear what's the object, but if you zoom in, if you look at this one, we can clearly identify the object. And also, this also improves us to see the other vegetation types as well as the health of the reed canary grass. After pre-processing the images, we used noise and haze reduction. We use a Fourier transformation approach in order to reduce the stripping, eliminate noise further, and other periodic anomalies in the raster imagery. As a result of that, we can see the spatial filtering, which eliminate the noise. If you look at the left-hand side image, it has more noise. But after the processing, after running the Fourier transformation analysis, we can see most of the reed canary grasses are visible now, even their leaf structure and their parameter, the distribution along the sides. Also, we can distinguish the shadow and non-shadow areas. For example, if I look at this area, probably I would think this is shadow area by looking at the initial image. But if I move to this, the correct image, probably it's a part of the reed canary grass, but it's more dense. That's why we cannot see the details. Also, the edges of the reed canary grass are clearly visible now compared to the initial image. Next, I would like to show some results obtained by applying the Wallis adaptive filter and the crisp filter. In this approach, first we have used a Wallis adaptive filter that adapts the contrast stretch to the region of the interest. Also, we have used a crisp filter, which allows us to sharpen the overall scene illuminance without distorting the thematic content of the image. For example, if you compare both images, left hand and right hand side, high contrast, so that we can identify the healthy and unhealthy reed canary vegetation. Right? For example, this area is most probably unhealthy, and this area is more healthy. Also, we can able to identify other species and their spectral features. For example, if I look at this area, and you can see how we can clearly see the vegetation type. So we can able to demarcate the edges on the reed canary grass and bushes. Most importantly, we could able to remove the effect of shadow. For example, if you look at this small ditch area, if I look at the initial image, it looks like a shadow or something underneath. We cannot see clearly, but after improving the image by image enhancing techniques, we can see that ditch is included different vegetation compared to the rest of the vegetation in this image. Once we have enhanced the image using previously discussed methods, now we use a non-directional edge enhancement approach. Basically, this approach was performed to enhance the edge features within the imagery using the pivot field to enhance the edges in both horizontal and vertical directions. If I consider the enhanced image, I can see the the vegetation types very clearly. Also, I can see some small vegetation types that I cannot see from the original image, but the enhanced image more clear now because you can see them. Also, the healthy versus unhealthy reed canary grass, that is our main focus. In here, we can see that it's more clear that's a healthy vegetation, and if I move to this area, that's more or less unhealthy vegetation. So as a result of that approach, we could be able to distinguish different vegetation types and their unique features or characteristics and difference between healthy and unhealthy reed canary grass. Also, I use vegetation indices. After performing all the image enhancement techniques, we created 6 different vegetation indices in order to see the small differences between various vegetation types. Also, these indices can highlight and enhance the differences that we cannot be observed in the original image. For example, I'm showing here 3 different vegetation indices I used in this study, including different vegetation index, green normalized diffraction index, and modified red edge simple ratio index. Layer stacking. After image pre-processing, we stack the enhanced multispectral image with 6 indices with the DSM data. So altogether now we have 11 bands for the image classification. The right-hand side shows the image with the 11 bands. It is more improved. Now we can see less shadow. All the vegetation types are clearly visible, and other features also clearly visible now. So in image classification, I performed unsupervised image classification approach. In this study, I have performed both k-mean and isolate, as I mentioned. So better results were obtained by k-mean. So initially we created 25 classes, and then later we merged them into 7 classes. So this is the image before the classification, and this is the image after the classification. Most of the features are clearly visible now with the 7 classes. Further, we can see the finalized classified image with 7 classes, as I mentioned earlier, including healthy reed canary grass, unhealthy reed canary grass, ground, vegetation type 1, 2, and 3, and also the shadow. On the right-hand side, we can see the results of the accuracy assessment. Importantly, we are focusing on the reed canary grass health and unhealthy. We can have 88 of producer accuracy and 95% of user accuracy for the healthy reed canary grass. Also, more importantly, if you look at the shadow and their producer and user accuracy, more importantly, we have lower producer accuracy for the shadow. This is because basically we could able to see the enhanced initial image, and we could able to pull up the spectral information which was identified as shadow in the initial stage. So this is a kind of a good output or good indicator about our technique is performing well. For the future work, immediate future, I'm planning to do temporal analysis of reed canary grass using multiple flights and analyze the effectiveness of the herbicide application with remote sense data. In future, perform supervised image classification and comparing the efficiency of different image classification approaches. With that, I would like to acknowledge Clean Water Services who initiated and support the research, especially John Goetz, Benjamin Portsman, and Brian Shepherd. And also I would like to acknowledge Mosaic Ecology who implement this project. These are my references, and yeah, thank you for the organizing committee and the all you attend the presentation.
Great, thank you, Sudeera.
Thank you.
We've got some time for questions if anybody has them. Matt Barker asks, which index most clearly showed differences in healthy versus unhealthy reed canary grass?
Generally, I tried around like 10 to 15 vegetation indices, but most prominently, I selected these three and another three. Like instead of these three, I have the NDVI and ratio vegetation index. All of them are good at identifying the reed canary grass, but if I need to specify which one is the better one, I had to do more analysis to pick best one. But overall, I use like 6 different indices out of 15 that I thought the most valuable things for the identifying the reed canary grass.
John Souter asks, how are you going to approach changes over time? Cumulative effects of treatments?
Yeah, so since we can gather the data from like temporally, the first thing we can do is we can separate the reed canary population from the image, and we can compare temporally how they change with the time. For example, if I compare the previous month with this month, and if I subtract the image, I can see whether it has an improvement of growth or decreasing of growth based on the treatment levels. So after the classification, we can do a lot of like binary classification approaches to address this kind of a question. And I think that would be the most appropriate way to address this one.
See, I don't see any in the chat. I had a quick question. I noticed you had sort of comparison of the different, or the supervised versus unsupervised classification.
Yeah.
Do you have any plans to compare the sort of remotely sensed classification to like an on the ground survey of some kind?
Yeah, that would be great if we can use the ground survey data to compare our remote sensing data to see how it performed with the actual data. Given that we have the RBG image, but that's not ground data, but it's another supplemental data to compare the actual data versus the classified image. But I would like to see some ground data, as you mentioned, to see the performance of the classification.
Bogdan asks, what was the most difficult task among all of the tasks that you did here?
Yeah, so the most difficult task is the computer space, because processing of these images are really consuming space, and the other one is running the different techniques take some time because I had to pick the best one. So it's like a trial and error approach. So spend a lot of time to bring the research to this level. So that's the most challenging part.
Hi, Sudeera.
Yeah.
I was just wondering if you think there's any way to reduce some of the image noise by designing the flights differently. Like if you accounted for angle of the sun or something, would you be able to reduce some of the striping before even pre-processing?
Yeah, exactly. So one thing we could do is enhance the overlapping, and as you mentioned, the pre-flights planning is also very important. But some of the data are already collected, so the only option we can do is go with the data and try to improve them after collecting the data. But yeah, as you mentioned, if we can plan the flights like high overlap areas and as you mentioned, like changing the different parameters, that would be great. Yeah, definitely I would agree with that.
Great, thanks, Sudeera. I think that's it
Yeah.
for questions, it looks like.
All right, thank you so much.
Yeah, thank you. Let's see, Kathy, do you want to go ahead and share your screen?
Yes. Can you guys see my video? Oh, okay. Okay, can you guys see it now?
Yes.
So, hi, thank you everyone for coming. And my research today will be titled Undergraduate Research Experiences, Impacts and Opportunities. And I'm doing this research with Dr. Pippi Larasati and Dr. Eric Hansen, which is from Department of Wood Science and Engineering. So first one, this research will be divided in 4 sections. The first one is why and how I applied to the MEP program. The second one, what I had to do for each project, and then how the experiences impact my personal growth and what opportunities lie ahead. So a little bit about me. My name is Kathy, and I'm an international student from Jakarta, Indonesia. I'm currently a sophomore majoring in forest engineering with a minor in forestry. And I'm also planning to take the GIS certification. Other than that, I'm also involved in the PERMIAS, or the Indonesian Students Association in OSU, as the Public Relations Officer, and I'm a research assistant. So MEP or Mentored Employment Program is eligible for undergraduate and post-bac students enrolled in COF undergraduate majors, and it happens annually. It's also available during fall, winter, and spring terms. And students work with members of COF faculty on research projects or field experiences. It's also a paid internship, and it provides limited amount of funding. And there are a few reasons on why I applied for the MEP program. The first one is I think it's a really good opportunity to work hands-on with faculty mentor on a project, either it's research, field, or teaching or extension experiences. Because I've always wondered, like, how do you apply this knowledge that you got in class outside in the real field? And it also provides experience that allows students to explore potential career paths and develop professional relationships, because you got to work closely with your mentor. And then how to apply. So you go to the OSU COF webpage and to the Mentored Employment Program. And this is like the deadlines from last year's. And if you're like me, which is a mentee or like students, you click on this link and then you would go to this website. And there's not much form that you need to fill. There's only 2. So you need to make a cover letter. And there is student services MEP interest form, which you only need to fill like what is your full name, your major, your expected graduation year, and also your top 3 choices of your projects, and then you're good to go. And this is and this is last year's project opportunities, and I signed up. Right now I'm working on this one. Does gender matter in a circular bioeconomy, and empirical and political perspective from forest sector practitioners. Not just that, I'm also working on Women's Network and assessment of research extension experiences for undergraduates. So I ended up working on 3 projects. So circular bioeconomy and gender. according to FAO, or the Food and Agriculture Organization, circular economy aims to maintain the value of products, materials, resources for as long as possible by returning them into the product cycle at the end of the use, while minimizing the regeneration of waste. The purpose of this project is to see if there's any connection between gender and CE. In this project, I'm involved in a research design, and my job is to collect CE government initiatives from each country. And I ended up making this table ranked by GDP per capita, also included with their gender inequality index, forest sector gross value added, forest area per capita, CE government initiatives, and the forest-based bioeconomy government initiatives. And other than that, I ranked around 107 countries. And then the next step that I will be doing is to type gender in every document to see if there's any relations or specific gender policy in the CE initiatives, then make a data analysis from that. The second project I'm working on is WOWNet, or the Women's Network. It's also, or you can say, Women Owning Woodlands. The Women Owning Woodlands project strives to bring topical, accessible, and current forestry information to woodland owners and forest practitioners through newsletters, blogs, events, resources, and personal stories. They support women in forest leadership, women who manage their own woodlands, and all who facilitate their stewardship forests, although it's not exclusive. So anyone who's interested in forest can basically join. And the purpose of the project is to identify impacts of women organization in the forestry field. And my job, I'm just included in the research design, which means I'm making transcripts out of the interviews. And my last project that I'm working on is the assessment of research extension experiences for undergraduates, or REEU. And the goal of the project is to assess successful past mentor-mentee relationships and how the program impacts mentees' career. And for this project, I'm involved in a data collection and data analysis. So I interviewed mentor and mentees, which is there's like 5 pairs of them. I also make transcripts of the interviews that I did, and then I'll eventually help with the data analysis, which means I will help write the paper. What are the impacts of the MEP to me? The main thing that I recognize that improves a lot after I joined the MEP program is my communication skills. I have been always struggling with talking to strangers, nor I am a big fan of doing presentations. But by doing a lot of interviews, as well as talking to someone who is more experienced and superior than me, definitely helped me gain confidence and improve my communication skills. Other than that, doing 3 projects as once, as well as becoming an officer for another organization, while being a full-time undergraduate, taking at least 16 credits every term, also taught me how to manage my time better and prioritize my work. I had to make sure that I am on track with my due dates and that my work are clear and concise enough to be understood by other people, so I won't have to spend more time on it, which also taught me knowing my limits. My mentor, Dr. Hansen and Dr. Larasati, has been very understanding, and they make it very clear from the beginning that they realize how many things I have on my hands, and to let them know if it's too much or if I needed more time, which I really appreciate. And another thing that I gain is networking. From MEP, I got to talk to a lot of experienced people in the faculty, something that I didn't have when I was just studying in class. After doing this research, I also realized that doing research or grad school is not that intimidating. Everyone is super helpful, super friendly, and they seem to love what they're doing. And doing research can be super fun when you're interested in the topic. Based on the interviews that I did with the mentees, a lot of them also said the same thing. Some of them even able to ask for recommendation letters to go to grad school, not just from their mentors, but also other faculty members who they got to work with aside from their original project. And opportunities ahead, of course, being able to speak here. Without doing my researches, I wouldn't be able to speak here. And then another opportunity that I'm really grateful with is being able to present at the my RE project at the SWSC, which stands for Society of Food Science and Technology. They held international convention every year, and it's just I feel honored to be able to present there, and it's something that I didn't expect to do when I was just in my sophomore year. And I think that's all I have for you now, and thank you. Do you have any questions?
All right, great job, Kathy. We've got a good amount of time for questions, and there's one in the chat already here from John Souter. He says, Kathy, how would you make the case to the dean to increase the amount of MEP positions and funding?
I think we haven't really assessed we haven't got to do a point of data analysis yet. So right now I'm just still doing the interviews and still making the transcripts, and then I'll be working on that with my mentors, Dr. Pippi Larasati and Dr. Eric Hansen, and then maybe I can talk to them about that. And yeah. Then second question.
Yeah, Steve Strauss asks, is your MEP nearly done, or how much longer do you have to finish up your projects?
My MEP program will be done in June, so a few more months, but maybe I'll be looking for other researchers after that.
Brianne asks, do you see yourself doing more research in the future after this program ends?
Definitely, yeah. Like I before taking this program, I thought research was really intimidating and it's like really hard. But after I got into it, it's something that I didn't expect that I would be able to do, and I would be really interested in doing other researches after this program ends.
Steve asks, do you feel comfortable analyzing the data that you're obtaining, or how will that data get analyzed, or how will you become comfortable analyzing it, I guess?
So this is my first research experience, and to be frank, I don't have a clear picture on how I'm gonna do that, so I'm gonna learn while doing it, I guess. And I'm assuming we're just gonna do the data analysis based on the interview transcripts that I'll be making, and I guess like other resources like papers.
I had a question. Oh, Eric Hansen says, I think learn by doing is the right answer. I think since there are a lot of grad students and faculty here listening, I was wondering if you could speak a little bit about sort of how you found the MEP program and how you were motivated to apply for it. Like, is there more that kind of faculty and grad students should be doing to reach out to undergraduate students or to advertise the program?
Yeah. So I know about the MEP program from Dr. Pippi Larasati, because she also happens to be Indonesian, and like she told me, there's like this great program which is a mentor employment program, and you'll be working closely with people in the faculty, and I think it's like a really good opportunity because I have zero experience in doing research, right? And to be able to have something close to work with you and give you directions when you have zero experience in that is really helpful. So and other than that, when I interviewed the REU participants, a lot of the mentors also mentioned that if you want, most of them get their positions by just emailing the teacher saying like, hey, I'm really interested in what you're doing. Can I do? I would really love to work with you if you're doing a research, or like they also applied on the programs from the newsletters they receive and the emails.
Oh, and Ariel points out to remember that the stats consulting program is available to anyone working with College of Forestry faculty. So that's a really good resource for analyzing your data also. Yeah.
Thank you.
All right, looks like that's probably it for questions. So thank you, Kathy.
Thank you.
Yeah. And let's see, it looks like we have about 5 minutes until our final talk for the day. So we'll just kind of sit tight until then. As always, feel free to use the extra time to fill out feedback forms for our speakers online. All right, Colby, are you all squared away?
I think so. Can you see my screen?
Yep. All right, so for our final talk of the day, we have Colby Parkinson here to share about environmental and individual level determinants of outdoor recreation participation during the COVID-19 pandemic. Yeah, take it away, Colby.
Thank you, Graham. So hello, everyone. As he mentioned, my name's Colby Parkinson. I'm a first-year master's student at Oregon State University in the College of Forestry. And today I'll discuss a study that analyzed the relationship between individual and environmental level factors that contribute to outdoor recreation engagement during COVID-19. I will then provide recommendations for green space and park managers and individuals seeking to improve personal or public health, as well as advice for future research. I'd also like to acknowledge the role of team members, particularly the additional authors Sharon Shen and Megan MacDonald, who played an integral role in conceptualization, study design, abstract submission, and every other important step along the way. To start out, I'd like to ask a quick question. Raise your hand or write in the chat if you have used the outdoors to help get through the pandemic. I can't quite, oh, I see some hands going up. So at least a few people. I know that I have too. So if that's not enough of a base to explain why we should study outdoor recreation during the pandemic, it's important to understand that there is evidence suggesting that outdoor recreation improves mental health. In particular, it seems to decrease stress and improve well-being. While the direct causal link may vary by study, design, and outcome variable, it generally seems to derive from increased physical activity and a sense of social connectedness. The factors that predict outdoor recreation outside of COVID-19 include nature orientation and values, as well as the qualities of nearby or accessible green spaces. While in general, demographics do not seem to provide a direct link between outdoor engagement rates, they do influence individuals' access and the quality of nearby green spaces. COVID-19, on the other hand, has changed daily life for over a year, with most counties in the US implementing changes in public health recommendations and orders in the March of 2020. Beyond inflicting harm through the disease itself, the pandemic has negatively impacted people's mental health, particularly affecting their anxiety and depression. People with pre-existing stressors are at even more risk, such as those with children with disabilities or chronic medical conditions. As you may have noticed, the benefits associated with outdoor recreation serve as possible candidates for buffering the negative impacts of COVID-19. With that in mind, changes in outdoor recreation have already been observed in the literature too, with many studies during the early phases of the pandemic suggesting that sedentary behavior increased and rates of activities such as hiking and biking decreased. Further evidence suggests that outdoor recreation, even among enthusiasts, has decreased during the pandemic as well. The research question that inspired this study sought to answer what individual and environmental level factors predict outdoor recreation engagement rates among adults in the US during the mid stages of the COVID-19 pandemic. The design of this study was cross-sectional, using a quantitative approach and a convenient sampling method via Prolific, which is a crowdsourcing software for sampling. While the data collected is non-probabilistic, it was also purposeful, with Prolific helping us make sure that our sample was representative of US adults based on gender, age, and race. The data was collected for the most part during the mid stages of the pandemic around January 2021. We studied environmental level factors that generally focus on access to nearby green spaces and the qualities of those green spaces that individuals visit. Additionally, at the individual level, we analyzed covariates that generally revolve around demographic and financial characteristics, as well as the importance of outdoor recreation to the individual. The dependent variable, or outcome variable in this study, is based on one item that asked, over the past month, how often did you recreate outdoors? Our results suggest that nearly half of participants recreated more than once a week on average. Generally, people who recreated outdoors were hiking, biking, or running. In terms of some additional descriptive characteristics of our sample's outdoor recreation behaviors, We examined the changes in outdoor recreation that individuals felt they had underwent during the pandemic, with evidence suggesting that fewer than 15% of people believed that they were recreating more than they were before the pandemic started, and nearly half of the sample feeling that they had recreated less. It is possible that many factors may influence these rates, though it is important to note that this took place during the winter whereas restrictions went in place in the March, so people may have been comparing the recreation rates from winter to spring. Additionally, during the winter, there were particularly high rates of infection to COVID-19. These factors may also extend to another question that we looked at, which was the current versus ideal amount of outdoor recreation, which showed that fewer than a quarter of people felt that they were recreating the proper amount during the pandemic. We plan to analyze the factors that may predict these forms of engagement in a future study. In terms of the current study, we ran a multiple regression model using 14 independent variables to study what may impact the frequency of outdoor recreation engagement. Overall, this model accounted for roughly 25% of the factors that predicted outdoor recreation. I would like to give you a few seconds to take a look at the beta coefficients and significance in this model, because I know that it is several variables. As you may notice, there are two factors that came back as statistically significant, which were safety and the importance of outdoor recreation. I'll discuss how these were measured. First, safety was measured by asking participants to rate their agreement with the statement, "There are safe green play spaces in our neighborhood for children and teenagers." Roughly 20% of the sample believed that to a degree they could not agree with that statement, or that they disagreed with that statement, meaning that maybe around a fifth of participants did not feel safe in their nearby green spaces. The importance of outdoor recreation, on the other hand, which was the most salient factor, was measured by asking, how important is outdoor recreation to you? Participants provided their responses using a 5-point unipolar scale from not important at all to absolutely essential. Most of the sample believed that outdoor recreation was moderately or very important, whereas fewer than 8% believed it was not important at all. The findings from this study suggest that people seeking to increase park use may benefit more from focusing on individual preferences over macro-level factors like aesthetics. This study may complement other research that has found that there is a baseline quality necessary for parks and green spaces to be used, but that additional quality may not be linearly related to further use. Campaigns to increase park use may benefit from instead focusing on making people appreciate outdoor spaces more. Public health campaigns, on the other hand, that want to encourage the safe use of green spaces should emphasize the value of green spaces. Currently, the CDC provides guidance for park managers and people interested in recreating outdoors, but provides little information on why it is safe during the pandemic and its potential benefits. The future direction of this research should focus on using a longitudinal design to best determine the causality between different factors. Additionally, a more rigorous examination of the importance of outdoor recreation to individuals, or an exploration of their orientation toward nature in general, would provide more depth of analysis and provide further evidence to support what is actually making people interested in recreating. Additionally, this research is premised on the idea that outdoor recreation decreases stress and improves physical health and social connectedness. So future studies should examine this relationship to ensure that it applies in the context of COVID-19. Thank you all for coming to my presentation. Please feel free to unmute or type any messages that you may have in the chat, or email them to me if you would like to discuss any of these findings.
Great, thanks, Colby. Let's see, Michelle Green has her hand raised. Let's see, Meg Krawczyk in the chat asks, do you think there could be interesting interactions among different factors in your model?
I do believe there could be, yes. In particular, I'll go back to the model itself. 1 second. In particular, these factors, like all of the factors regarding green space, were inspired by other work. But I can imagine that perhaps someone who thinks that there's good facilities and a park is aesthetically pleasing may also feel more safe in that space. Additionally, the walking distance and access are approaching access to the outdoors from a different approach, where one is asking if people feel that they have access and the other is more an objective measure based on how far they must walk, but they could be interacting as well. And in terms of at the individual level, there are two factors looking at financial security and household income that could certainly have an interaction, but we wanted to parse these out because financial levels across the US will vary. So, you know, a certain degree of income in California is going to have a different impact versus a state that on average has like a lower income. So we wanted to make sure that we were addressing it from both angles.
I was wondering about you mentioned that the survey was conducted in the winter. And I was wondering if you think that that has a big effect on this finding that safety is a really salient point for a lot of people. I know that in the winter, I tend not to get out as much just because I don't really want to be out in the woods, like riding my bike at night, especially with the cougars around here. So I guess I was wondering if you thought that might influence your results, like the seasonality.
Yeah, we were a little concerned about that. We figured that it was better to try to collect the data in as quick of a time as possible, which is one of the advantages to using prolific, so that at least between subjects, it was as accurate as possible. But I agree that certainly the best next direction for this research would be to conduct it likely around this time of year or early summer, and then draw comparisons between the groups to ensure that this wasn't a factor. I will say it's going to be hard to compare because, as I mentioned too, in the winter, rates of COVID-19 were different. And, you know, there's several other factors that are going to be seasonal. So it's important to view this as a snapshot during that time of year. And it may, you know, differ across different timings.
Let's see. I'm not seeing any more. I had one other question. I noticed that, you know, you had this slide showing 3 bars or 5 bars where a lot of people were not getting out as much as they wanted to. Yeah, or I guess the one after that is the one I was thinking of. And I guess I was thinking about a lot of in Crystal Colden's talk this morning, she mentioned that during the pandemic, a lot of folks are recreating more in national forests and things like that, and that maybe is leading to more fire risk in some ways. And that kind of seems like it contrasts a little bit with your results. And so I was wondering how, you know, where's that nuance, or like how does that square in your mind?
Yeah, I mean, in my mind, and this is purely based off of, you know, this isn't scientific. This is just based off what I've read, but I think that maybe sites that are going to attract a lot of people, like national parks and things of that nature, have seen a substantial increase in visitation. And I think that there's even some literature supporting that, whereas recreation in general has decreased. And so I think that it's particularly like an important question here is that there's actually evidence from an author at Penn State he showed that outdoor recreation among enthusiasts has gone down during the pandemic, or at least it had over the summer of 2020. But it is possible that maybe outdoor recreation rates have increased among non-enthusiasts. And so that may mean that the behaviors of outdoor recreation are different, because if people are new to outdoor recreation, they may be more likely to go to the more popular places like a national park or a national forest nearby, as opposed to people that are, you know, more frequent hikers in their neighborhood or bikers in their neighborhood.
Well, thanks.
Yeah.
All right. All right, well, I'm not seeing any more questions in the chat or any more hands raised, so I think we can say thank you to Colby. And I'll say thank you to everybody else who's participated either as a presenter or giving feedback or even just attending these sessions. Thank you for making WFGRS possible this year. It's been really, really great to see all the cool research that's going on in the college and get to see that shared. As one final note, even though I'm sounding like a broken record, please do fill out those feedback forms for our presenters if you can. There are links on the website underneath each of their names. And with that, I'll sign off. Thank you, everybody.